Good afternoon, students. Welcome back to the YouTube channel, The Guide, The Knowledge Hub. This is Ramesh, Faculty of Biology. Please subscribe, like, and share the YouTube channel, The Guide, The Knowledge Hub. Dear students, today I am going to explain the topic cell membrane or plasma membrane. Cell membrane or plasma membrane. Okay, listen carefully and follow me. So pay the concentrate all of you. So dear students, you well known about cell. Actually, cell is the basic unit of life. Says or no? Cell is the basic unit of life. So the group of cells together and formed tissue. The group of tissues together and formed organs. The group of organs together and formed organ systems. The group of organ systems together and formed one organism, means some life. Okay, so that is the importance of cell. That's why we are telling cell is the basic unit of life. So we have learned in earlier classes about cell. Okay, today I am going to explain the topic cell membrane, means plasma membrane. Dear students, in your earlier classes you have already studied that cell membrane. So it is the covering of the animal cell, and plant cells also there. So in plant cells there is another layer, so present over the cell membrane, that is called a cell wall. So the cell membrane is present in the animal cells. Along with cell membrane, means plasma membrane, there is another layer in plant cells, that is cell wall. So here, observing the cell membrane, how we observe the cell membrane, how we will observe the cell membrane. For this, we need to conduct an activity, but here there is no apparatus to conduct of this activity. So this is rheolith. What is this? Rheolith. Why I need to select this rheolith? So I wanted to observe the cell membrane in this rheolith. So dear students, first of all, take a rheolith, tear the leaf in a single stroke. So we have to tear in a single stroke. So like this. So I have to tear it. So we have to tear this rheolith in a single stroke. Okay. So this is rheolith. So then observe it against the light. So observe it against the light. So take a small piece of leaf peel with the light color, with the light colored this is transparent portion. Okay. So put it on a slide and put a drop of water. So we need some apparatus, we need some materials. So we need some required materials to do this activity. So I need to place this rheolith, transparent piece of leaf, rheolith, on the slide. Then I wanted to add one drop of water. So after cover it with cover slip, we were known about this preparing slide. So under observe light portion of leaf under the microscope. So then, while you are observing under the microscope, then we are the responsible to observe very keenly. So we need keen observation under the microscope. So while you are observing under the microscope, you have to draw the diagram of what we have seen under the microscope. So now put 1 to 2 drops of dilute salt solution on this membrane and leave it for 5 to 10 minutes. So here I told you, please add salt solution on this peel of rheolith. So salt solution, how we prepare salt solution? Yes, we know. For preparing salt solution, take 50 ml of water and add 1 or 2 teaspoonful of salt in it and stir it well. That is salt solution. So after added the salt solution, the picture is looking like this. So you know about this. So real leaf peel cells with membrane. So this is the rectangular shaped structures are visible. They are appeared on this. So on under this slide, okay, and under the microscope. So here a round bound structures are there. They are the nucleus. You may observe. So where these nucleus are present in this. So the nucleus is present in the corner of the cell. Each and every cell contains corner of the cell. In plant cells, the nucleus is present in the corner of the cell between cytoplasm. Okay, this is about real leaf peel cells with the membrane. The structure is looking like this. Once all of you look at carefully. Are you observe clearly? The nucleus is present in the corner of the cell. So that is the real leaf peel cells. So, dear students, here when we put salt solution over the peel of real leaf, so the water is present in the cell. So why we are adding salt solution in this? Because of the water present inside the cell, real leaf, those cells which comes out when we added salt solution. Okay, it results in the shrinking of the cytoplasm along with the cell membrane. So the outer boundary of colored area is actually the cell membrane or plasma membrane, which becomes separated from the cell wall. Okay. However, we can observe the structure of cell membrane only through an electronic microscope. Cell membrane is the flexible and is made up of with the proteins and lipids. So once look at this picture, real leaf cells. These are the plasma. So outer cell. This is outer covering is plasma membrane. So these are the cell walls, and so a pinker structure there now, that is sunken cytoplasm between the cell. That is the cytoplasm. So cell membrane is the outermost layer of the cell. So what is the importance of cell membrane? So the cell membrane is the outermost layer of the cell that separates cytoplasm from the external environment. Okay, this is also known as plasma membrane. So once again I am repeating. So what is the role of cell membrane? It separates. The cell membrane separates cytoplasm from the outer environment. This is also called as plasma membrane. So the cell membrane defines shape. Cell membrane defines the shape and size of the cell. So it encloses the cytoplasm and protects from the external environment. What is the role of this plasma membrane? The plasma membrane protects the cytoplasm from the external environment. The internal environment of this cell is different from the outside, external environment. Okay, so the cell membrane plays a crucial role in maintaining the balance. So the balancing of the nature of the cell is maintained by the cell membrane. So any substance entering or leaving from this cell can do so only through this membrane. So for example, so I told you already, we have a multicellular organisms. We have a multicellular organisms and some unicellular organisms also there. So single-celled organisms means protozoan organisms. The single-celled organisms means protozoan organisms are amoeba, paramecium. These type of organisms can perform. They are the single-celled organisms. The single cell can perform all the life activities in that single cell organisms, unicellular organisms. One single cell can perform all the life activities. But here we are made up of, animals are made up of, plants are made up of multi-cells. So our cells, our multi-cells, number of cells can perform various functions. So the multi-cells, number of cells are present in our body. All these cells can perform in different activities. Each and every cell can perform in different activities. So here also this cell, so these cells are allowing substances into this, and leaving some substances through this cell. Okay, the cell membrane playing a very key crucial role, that is it's allowing some particular substances. So the uniqueness of this membrane, so it is lies on the fact that is that it does not actually the plasma membrane, this is the plasma membrane does not allow unwanted substances through this. So the plasma membrane will allow some particular substances through this. That's what we are calling semi-permeable membrane. Once look at this diagram all of you. Once look at carefully all of you. Clearer? Okay, this is. So next look at carefully. So the exchange of substances, I told you already, some substances, some particular substances allowing through this cell membrane or plasma membrane, and unwanted substances are not allowed. It is stopped. Okay, the exchange of substances through this cell membrane happens very selectively. So hence it is known as selective permeable membrane. So it is allowing some particular substances only through this cell membrane or plasma membrane. That's what we are calling semi-permeable membrane. So this characteristic of membrane explains to the control the exchange of substances between the cell and its external environment. Okay, so the reduce this structure, the cell membrane structure of the membrane was studied only after the advance of electronic microscope, for example. So how can we study this about cell membrane? So we have to know the more than details of this cell membrane under the microscope only. So we are unable to observe normal microscope. We are unable to observe the simple microscope. We have to observe, we are the responsible to observe. We have to observe keenly under the electronic microscope only. So we well know about in the 1950s, they observed under the electronic microscope. So the chemical studies on the cell membrane, especially in human beings' red blood cells, enable the scientists to deduce the possible structure of the plasma membrane. First of all, the scientists are worked on human beings' red blood cells and they decided the structure of the plasma membrane. So these studies showed that cell membrane is composed of lipids and proteins. So some lipids and proteins are arranged on a bilayer. So also the lipids are arranged. The lipids are arranged with the membrane, the polar head. So towards the membrane. So there is a hydrophilic membrane. So having a tendency. So the hydrophilic tails towards the inner part of cell membrane. So this ensures that the non-polar tail of saturated hydrocarbons is protected from the aqueous environment. So the lipids compounds, the proteins compounds of the membrane mainly consists of phosphoglycerides. You know already the glycerol containing materials, phosphoglycerides. So later the biochemical investigations clearly revealed that cell membrane also possesses proteins and lipids, various considerable in different cell types. So in human beings, the membrane of erythrocytes, the red blood cells there means erythrocytes. The erythrocytes have approximately 52% of proteins and 40% of lipids. So depending on the location and easy of extraction of membrane, proteins can be classified as integral or so actually these are classified integral and peripheral. So the peripheral proteins lie on the surface of cell membrane here. So thus these are the